Good day everyone. Today, my group will be presenting our topic, phenylketonuria, a rare and one of congenital disease. And this is the outline for today's presentation. So, what is phenylketonuria or PKU? PKU is an autosomal recessive disease caused by mutation in the phenylalanine hydroxylase gene. This gene produces the PAH enzyme, which is responsible for converting the amino acid phenylalanine into tyrosine. When the enzyme is deficient or absent, phenylalanine accumulates in the blood and body tissue to toxic level. The excessive accumulation of phenylalanine is particularly harmful to the brain. Elevated phenylalanine level can disrupt normal brain development, impair neuronal growth and myelination, and hence it will lead to the neurocognitive problem if left untreated. As a result, untreated PKU may cause severe intellectual disability, developmental delay, seizures, and behavioral disorder. However, this complication can largely be prevented through early diagnosis and proper management. Today, newborn screening program allow PKU to be detected shortly after birth. Once diagnosed, patient can be managed through a phenylalanine restricted diet, specialized medical formulas, and in some cases, pharmacological therapies. Early intervention is essential to maintain normal neurological development and improve quality of life.
Phenylketonuria or PKU is autosomal recessive disorder classified as aminoacidopathy. It is commonly caused by mutation in the PAH gene, which encodes the enzyme phenylalanine hydroxylase. This hepatic enzyme is responsible for converting phenylalanine into tyrosine, a key step in amino acid metabolism. Phenylalanine hydroxylase or PAH is the rate-limiting enzyme in this pathway. It requires three important cofactors, tetrahydrobiophyrin, also known as BH4, molecular oxygen, and iron. BH4 binds to the catalytic domain of PAH and is essential for the enzyme to carry out the hydroxylation of phenylalanine to tyrosine. When PAH activity is reduced or lost, phenylalanine accumulates in the blood, leading to a condition called hyperphenylalanemia. At the same time, toxic metabolites such as phenylpyruvate, phenylacetate, and phenylate build up. This is due to phenylalanine is not converted to tyrosine. The body also develops a relative tyrosine deficiency. Elevated phenylalanine crosses the blood-brain barrier, competitively inhibit the transport of other large neutral amino acids including tyrosine and tryptophan. This reduces the availability for neurotransmitter synthesis, leading to lower level of dopamine, norepinephrine, and serotonin, which are critical for normal neurodevelopment. Infants with PKU appear normal at birth. However, without treatment, symptoms develop over the first month of life. These include characteristic master odor of the skin or urine, irritability, tremor, seizure, developmental delay, microcephaly, and neurocognitive impairment. Due to tyrosine is precursor for melanin, it reduces tyrosine level lead to hypopigmentation of the skin and hair. The musty odor is caused by phenylatic acid, a phenylalanine metabolite that accumulates and is secreted in sweat, urine, and skin secretion.
CRISPR-Cas9 is a gene editing technology that acts like a molecular scissors to cut and repair specific mutated genes, and it has potential to treat PKU by targeting the faulty PAH gene. However, there are still challenges. Delivering CRISPR into liver cells is not very efficient, and off-target edits may cause unwanted genetic changes. In addition, not all cells get corrected, so PKU may still persist. The mechanism of CRISPR-Cas9 in PKU involves targeted gene editing of the PAH in liver cells. A guide RNA directs Cas9 to the faulty PAH gene, where it cuts the DNA and creates a double-strand break. The cell then repairs the DNA using a template or natural repair mechanism like NHPJ or HDR, restoring PAH function so phenylalanine can be broken down properly.
Now, I will explain the second biotechnology solution for treating phenylketonuria or PKU. One promising approach is S1MB1618, which is an engineered synthetic probiotic developed from Escherichia coli in the cell. Its main function is to degrade excess phenylalanine or pH directly in the gastrointestinal tract before it can be absorbed into the bloodstream. S1MB1618 uses two metabolic pathways, PAL and LAAD, to convert phenylalanine into harmless byproducts. However, there are some challenges. The expression of PAL and LAAD can slow down bacterial growth. In addition, large-scale cultivation is difficult because bacterial growth requires oxygen, while PAL is expressed better under anaerobic conditions. This slide shows how S1MB1A degrades phenylalanine through two different pathways. The first is the PAL pathway, where phenylalanine is converted into tranexaminate. This process is enhanced by the PHEP transporter, which increases the uptake of phenylalanine into the bacterial cells. The second is the LAAD pathway, which converts phenylalanine into phenylpyruvate. Unlike PAL, LAAD is active in oxygen-rich environments, such as the proximal small intestine. These two pathways improve the efficiency of phenylalanine degradation in different conditions within the gut. This slide explains how SYNB1618 reduces blood phenylalanine levels through enteral circulation. Phenylalanine from the blood naturally moves back into the gut. SYNB1618 degrades this phenylalanine before it can be reabsorbed, helping to lower blood pH levels. This approach is effective for both dietary phenylalanine and phenylalanine returning through enteral circulation. It also works regardless of protein intake and has shown positive results in PKU mouse models and non-human primates. Overall, SYNB1618 offers a promising bioenergy strategy for managing PKU by reducing phenylalanine directly in the gastrointestinal tract.
In conclusion, phenylketonuria or PKU is a genetic disorder that disrupts phenylalanine metabolism, leading to serious health complications if untreated. While early diagnosis and dietary control remain crucial, emerging biotechnological approaches such as CRISPR-Cas9 and synthetic probiotic show promising potential solutions in the future. This is all the references list. With that, thank you for listening to our presentation.